Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were discussing about neurobiology, in which we have covered electrical properties of neurons. So, all of the basics being covered, today we are going to learn about neuromuscular junction. So, neuromuscular junction is a well studied example of directly gated signal transmission. Means whatever signal it arrives at the in the form of ligand which binds to the receptor, and this chemical signal is directly converted to an electrical signal which is passed down from the postsynaptic membrane. So neuromuscular junction is relatively simple and it is quite accessible for experimentation, and that is why it is quite it is well studied. So before learning about complexity of synapses that are present in our central nervous system, we will learn about the synapse that is formed at the neuromuscular junction. This synapse is formed between a motor neuron and a muscle fiber, and that is why it is known as neuromuscular junction. So this is very simple and it is very accessible for experimentation because the muscle fiber is quite long and multiple electrodes can be inserted to measure the action potential or any potential changes that are happening in the muscle fiber. And that is why we are going to learn about neuromuscular junction as an example of directly gated signal transmission. So the basic signal transmission is something like this. The signal arrives at the motor neuron. Okay, so motor neuron has to give command to certain muscle, and it is forming a neuromuscular junction. So this motor neuron, at the end of axon terminals, it is going to release neurotransmitter. Neurotransmitter is here a chemical signal. So this neurotransmitter is released from presynaptic terminal, and it opens a single type of ion channel on the postsynaptic membrane. So it will open, so let's say that the neurotransmitter that is being released by the presynaptic terminal is the acetylcholine. So acetylcholine will only, it will only open the single type of ion channels, the ion channels which are going to receive the acetylcholine. So we refer them as the acetylcholine receptors that are present on the muscle fibers. So in this case, the acetylcholine receptors that are present on muscle fiber, they are nicotinic acetylcholine receptors. So there are two types of ACH receptors, the nicotinic acetylcholine receptors and muscarinic acetylcholine receptors. The nicotinic acetylcholine receptors, they are ionotropic, while muscarinic, they are metabotropic. We will learn about those in quite detail, what are the differences and all in some other lecture. For now, it's what I want to convey is that whatever neurotransmitter will be released from the axon terminal, that is going to open a single type of ion channel which are present on the muscle fibers. So that makes the signal transmission and the study quite easy. Okay. So this particular structure that is formed by the motor neuron, when the motor neuron it integrates, it branches, the axon of the motor neuron branches and forms connection with the muscle fiber. This structure, whatever is formed, is known as end plate. So end plate is the junction or the structure which is formed by the motor neuron when the axon of the motor neuron splits into multiple branches and whose ends form a synapse with the muscle fibers. Okay, so the myelination is loosed at the axon terminals. So here you can see the myelination is ending. So even though the myelination ends, the end axon terminals here, this is the zoomed image of the end plate. So this is the end plate. Okay, this is the end plate region. Here you can see this is the end plate region. So the myelination is loosed at the axon terminals, but the Schwann cell, there is a single and thin layer of Schwann cell which is known as Schwann cell sheath, which is wrapping or covering the axon terminals. Okay, so we refer it as the Schwann cell sheath. So there is no myelin sheath, but there is Schwann cell sheath. Schwann cell sheath is a single layer of Schwann cell which is wrapping itself around the axon terminals. So axon branches into it splits into fine branches, and they at the end of those branches they form a swelling. Okay, this swelling here you can see the cross section of the swelling. That swelling is known as synaptic botons. Okay, let me show you. Here you can see that those swellings that are formed at the end of the axon terminals, they are known as synaptic botons. Okay, so then let's take a look at the zoomed image of the end plate region. So this is showing the whole schematic, the detailed structure of the end plate region. So this is the end plate region. Okay, so the axon terminal, that is going to form a swelling, okay, a protrusion, okay, and this swelling, so this swelled part is known as synaptic button, and it is going to form a connection with the particular region of the muscle fiber, and that region is known as junctional fold. Okay, so this you can see those folded regions. Here you can see those folded regions. Okay, so this is the junctional fold. So junctional fold is the region of the muscle fiber that is forming synapse with the synaptic buttons. Okay, so this is the synaptic button. It has mitochondria, it has these vesicles, okay, which contains the neurotransmitters, and those vesicles are known as synaptic vesicle. In this case, the neurotransmitter that is present in those synaptic vesicles is the acetylcholine. Okay, so the fusion of the synaptic vesicles happens in a particular area which is present on the presynaptic membrane, and that area is referred as the active zone. Okay, so active zone has particular characteristics, like on the active zone there is the voltage gated calcium ion channels are quite high. The concentration of those ion channels is very high. We'll know why. Okay, so the active zone is the zone or the region of the presynaptic membrane where the synaptic vesicles are clustered. Okay, they are, see, you can see these are the active zones. These are the active zones, and at the active zones, here you can see the synaptic vesicles are clustered. Okay, so synaptic vesicles are clustered here, and they are going to fuse on those particular zones. So that they are known as active zones because the action, whatever action is going to happen, that is going to happen through this zone. Okay, so these are the synaptic vesicles containing acetylcholine. This is the presynaptic membrane. Okay, and here the gap between the presynaptic membrane and postsynaptic membrane is referred as synaptic cleft, which is almost 100 nanometer wide. Okay, so this is the postsynaptic membrane. So the postsynaptic membrane is a part of the muscle fiber, while presynaptic membrane here is it is the part of the nerve end. Okay, the axon terminal. Okay. Sometimes the postsynaptic membrane could be a part of endocrine cells, or it could be a part of other neuron. Okay, so it could be part of the dendrite, the branches of the cell body. Here it is the part of the muscle fiber. Okay, so we learned about postsynaptic membrane, active zone, and calcium ion channels. They are present on the higher concentration on the active zone. So these are the calcium ion channels. This are the calcium ion channels. Okay, so those are the active zones. So then there's a thin layer of in between the muscle fiber and nerve terminal. Both of them, they are connected by a thin layer of connective tissue, and that is known as basal lamina. Okay, so basal lamina is a thin layer of connective tissue. Okay, so this connective, so it has, so as it is a layer of connective tissue, it has collagen, the extracellular matrix components like collagen and glycoproteins. Okay, so this basal lamina is, the presence of basal lamina is quite important because unlike cellular membrane, unlike any plasma membrane, lipid bilayer, this is freely permeable to small organic compounds. So this is permeable to neurotransmitters. So through this, neurotransmitters, they can pass through this basal lamina because of the permeability. So the both the presynaptic membrane as well as the postsynaptic membrane, both of them, as they have the basal lamina in between, they secrete their proteins on the into the basal lamina. Okay, so whatever neurotransmitter are released from the presynaptic membrane, that will be that will be released into the basal lamina. They will cross basal lamina and then bind on the receptors that are present on the postsynaptic membrane. as well as whatever proteins are being secreted, like the enzymes that are being secreted from the postsynaptic membrane. The enzyme like acetylcholine esterase, which hydrolyzes acetylcholine and breaks down the acetylcholine into acetate and choline, so that overstimulation doesn't happen. So the thing is that when the neurotransmitter is released after the synaptic vesicle fusion, so that neurotransmitter will be released, that will travel through the basal lamina and that will bind to the receptors that are present on the postsynaptic membrane. Once they bind to this receptor, the ion channels, so these receptors, the acetylcholine receptors, they are basically have two parts, the acetylcholine binding part and the ion channel, the pore forming part of the protein. So these are the transmembrane proteins, right? So the ion channel will open and it will allow the ion influx that is going to change the voltage of the postsynaptic membrane, and that creates the potential, the change in the membrane potential, that is known as end plate potential. We'll learn about end plate potential as well, but right now I wanted to focus on the thing that this is how the chemical signal that is arriving on this postsynaptic membrane, that is changed into an electrical signal. Okay, so once this is done, now you have to remove the acetylcholine molecules that are present in the synaptic lift. And for doing this, the postsynaptic membrane releases an enzyme known as acetylcholine esterase, ACH esterase, acetylcholine esterase. Okay, I'm writing the short form here. So this acetylcholine esterase, this hydrolyzes acetylcholine into acetate and choline. So this inactivates the signaling and this ensures that overstimulation won't happen. So once the signal is reached to the muscle fiber and muscle has contracted and it has done its job. Now you need to stop the signaling, and this is where the enzyme plays a role. So whatever proteins that are being they are being released from the postsynaptic membrane or the presynaptic membrane. What does presynaptic membrane release? It releases neurotransmitter. And what does postsynaptic membrane release? It releases acetylcholine receptors. So whatever they release, they are that all those contents, they are released into this basal lamina. Basal lamina is a connective tissue layer, okay, and this basal lamina is permeable, and through this, the whatever molecules are being secreted, they pass. So basal lamina, so that is why the basal lamina has an important role into organizing the synapse by aligning the presynaptic botons and the postsynaptic junctional folds. So it maintains a quite organized structure. Okay, so this is why basal lamina is quite important. So these are the junctional folds here you can see, and on the junctional folds voltage gated sodium ion channels are present, because once so what happens is that once the neurotransmitter acetylcholine binds to those acetylcholine receptors, those green ones are the acetylcholine receptors, while these situated at the bottom of the junctional folds, these are the voltage gated sodium ion channels. And what is the difference between both of them? Acetylcholine receptor channels, these are the ion channels which are permeable to any cation. So they are permeable to sodium, they are permeable to potassium, they are permeable to calcium as well, but here we are just focusing on sodium and potassium. So they are non-specific, they are not selective, while voltage gated sodium ion channels, they are very selective to the ion that they are going to pass. So they only pass the sodium ions. Okay, and what is the next difference between both of them? The acetylcholine receptors, they are ligand gated ion channels. Okay, so the ligand acetylcholine binds to those receptor channels and it opens the ion channel. Okay, well, this voltage gated sodium ion channels, as the name suggests, this is a voltage gated ion channel, means the change in the change in the voltage of the membrane is sensed by this protein, and then the gate opens, which allows the ion influx. We have learned about all of those things in the ion channels lecture. So if this is not clear to you all, please refer to that lecture. Okay, so once the acetylcholine binds to those receptors, now it is going to open the ion channel, the ion channel, the pore that is formed by this receptor, and now this will do the influx of cations. So once the cations arrive in the intracellular matrix, means in the cytosol, the negatively charged cytosolic part will convert it into positive. Okay, so the charge separation is reduced. This causes the depolarization of membrane, and that depolarization changes the membrane potential, and this change in the membrane potential creates the end plate potential. So if you have learned about graded potential and action potential, you will be able to answer this question. So what kind of potential is the end plate potential? Well, end plate potential is the graded potential. It is the graded potential because it is, it is it is being generated, and the amplitude of that potential depends on the number of the receptors, number of acetylcholine receptors that are present on the junctional folds. Okay, so this is created locally. This is the passive potential that is generated, and once this end plate potential reaches to certain threshold value, okay, let's say that it reached to the plus 55 millivolt, and this is the threshold value for the action potential. So once this reaches to a threshold value, then it will suddenly open multiple voltage gated sodium ion channels. So now there will be influx of sodium ions through those voltage gated sodium ion channels, as well as through the ACH receptor. There will be influx of sodium and potassium, and here the influx of sodium will be seen. So this is going to increase this there will be sudden, there will be sudden increase in membrane potential, and that will create the action potential. Okay, so what I wanted to convey here was, let's write the things step by step. So neurotransmitter is released into synaptic cleft. Okay, so the acetylcholine will bind to the acetylcholine receptor. Okay, so once acetylcholine binds to the receptor, that is going to open the ion channel. Open ion channel means the pore that is formed by the acetylcholine receptor itself. So this opening will do the influx of cations. Because acetylcholine receptor ion channel, this is not selective for particular cation, but it is only going to do the influx of cations. It won't let any anion pass through. Okay, it is going to do the influx of cations. So this will, momentarily change the membrane potential. So there will be small deviation in the membrane potential, and that is known as end plate potential. Well, end plate potentials that are created at the neuromuscular junctions, they are not really small. They are quite, they have a significant value. Okay, so this creates the membrane potential change, which is known as end plate potential. So this is a graded potential because it can be summed depending on the how many SEH receptors are opening, how many ion channels are opening. Depending on that, the the amplitude of this potential changes, and that is why this is a graded potential. So once this end plate potential is generated, and let's say that the value crosses the threshold level, the threshold level in each case that can differ, and we are assuming that the threshold level here is plus 55 millivolt, and so once it crosses this threshold level, it is going to open so voltage gated sodium ion channels. Okay, so now because that it is, it has this, the voltage, the membrane potential has passed on the certain required value. So that is going to, that voltage change will be sensed by those ion channels, and they will open, and they will do the influx of sodium ions. Now this will be, this will be sudden. Okay, and a lot of sodium ion channels will open, and this is going to create a sudden spike in the membrane potential. So the value of membrane potential will increase suddenly, and that will create action potential. That value change is known as action potential. we have already discussed this in very detail, in great details in previous lecture. So please refer that. So once this action potential is generated, now this action potential has the capability to travel across the long distances. And now this action potential, it is generated in the muscle fiber, and that is why it is known as muscle action potential. So this muscle action potential is going to result into the muscular contractions. Okay, so this is how the signal transduction happens in the end plate region. So the end plate potential, this was first studied by this was first studied in 1950 by Paul Fatt and Bernard Katz. Okay, both of them, they studied this end plate potential using the intracellular voltage recordings. Okay, so we already know that the introducing electrodes, the electrodes in the muscle fiber, it is quite easy, and that helps in the experimentation. So they used the intracellular voltage recording, and they used a drug known as curare. Okay, and this is a very important drug. By the way, this is a mixture of plant toxins. Okay, curare is mixture of plant toxins, and it was used by South American Indians. For they used to apply this curare on the arrowheads, and those arrows were used to hunt down their prey. So they used to that used to paralyze the prey. So curare it was the paralyze it was used as the paralyzing agent while hunting. Okay, so this curare, this plant toxin, the mixture of plant toxin, this was used by those scientists, the Paul Fatt and Bernard Katz, to isolate the end plate potential. So they isolated this end plate potential by reducing the postsynaptic potential below the threshold that is required for action potential. So once this end plate potential, it is decreased to that value. So if it is decreased below threshold, so curare helps them in creating sub-threshold end plate potential. So due to the subthreshold end plate potential, action potential won't be generated. Okay, so that helped them in the study. And when they studied those potential generations, they found out that this excitatory postsynaptic potential, by the way, the acetylcholine that is going to excite the muscles, that is why it is known as excitatory postsynaptic potential. So this end plate potential is the excitatory postsynaptic potential. Well, we discussed about those types in the last lecture. So if you don't know, please refer to that lecture. So excitatory end plate potential that is created by the end plate potential, that was that they found out that it is the largest. Okay, at the end plate compared to whatever is formed. I already told you that at the end plate potential, whatever potential is formed, it has a significant value, but it decreases. So they found out that this decreases progressively over a distance. Okay, so this also points out that it is a graded potential, because only graded potential shows the decremental conduction. Okay, while action potential, it shows either the continuous conduction or the solitary conduction. So this again confirms the point that it is the graded potential. And I told you when we were discussing about graded potential that depending on where and when it is generated, it has different names. So it's like the graded potential here has the name end plate potential. It can be also be called receptor potential or generation potential. Well, here it is the end plate potential. So they found out that this potential is largest at end plate, and it decreases progressively over certain distance. So this end plate potential, this is generated by the inward ionic current. So the movement of the positive ions, movement of cations is inward, right? So this is the direction of movement of the cations, and that is why this they found out that the current is inward. Okay, so whatever current is generated, that is inward current. So this inward current, this is confined to end plate region, and it spreads. So this inward current is confined and it spreads over the distances through the end plate region, and it dies out eventually. Okay, so they found out that it dies out, and then they also found out this current, it rises rapidly but dies slowly. This was also a significant observation made by them. So this made them to conclude that the end plate potential, it is the graded potential, and it travels passively, because only end plate, the graded potential, has the property to die out over the distances. So the whatever conduction was there, that is passive conduction. So it is not maintained by it is not maintained actively by opening the voltage gated ion channels. It happens in the case of action potential, and that is why we are referring this as a graded potential. So these are the recordings that were done by them. So here you can see the membrane potential. This is the end plate potential, okay, above threshold value. Once it crosses the threshold level, it is going to generate action potential. Okay, that is why the dotted line is showing the end plate potential, because once it crosses the threshold value, it is going to generate action potential. So this will be recorded as this will be recorded in whenever a stimulus is arriving. But when you use the Curer, this toxin, this is going to, what does Curer do? Actually, Curer, just like alpha bengarotoxin, this is also a toxin. We'll discuss about alpha bengarotoxin in some other lecture. So Curer, this binds to acetylcholine receptors. Okay, so as this Curer is bound to acetylcholine receptor, now acetylcholine cannot bind to those receptors, and this won't result into the opening of ion channels. So as the gate doesn't open, there will be no ion influx, and the signaling will be reduced. So using a certain concentration of Curer, they ensure that a few receptors will be blocked and a few are open. So this will create current. So normally a stimulus should generate an action potential, but the stimulus of same strength, now in the presence of Curer, it is going to generate end plate potential, which is below the threshold level, because some of the receptors, they are blocked by Curer. And this also confirmed that the conduction of the ion current was passive, and it decreased over the time. So this was the recordings that were made by the scientists. Well, this is a picture. Okay, this is a picture when this is how the receptor, the acetylcholine receptor, looks like when you see that, when you visualize it from above. Okay, so this is the low resolution electron micrograph of the acetylcholine receptor from side view. So here you can see it is the lipid bilayer. This is the lipid bilayer, and this is the acetylcholine receptor. And through this crack, the acetylcholine molecule slips through, and it binds to the receptor. Well, if you are wondering where this image are from, so I have taken all of the image that I'm displaying in this lecture. All of them, they are taken from the book known as Principles of Neural Science by Eric Kandel, and it is quite interesting book. And whatever I am explaining today, that is all that is documented in that book in quite details. So if you want to know more about it, you can refer to that book as well. So this is how the receptor looks like. Well, this is the study done by, again, fat and cats. This is how they, when they measured the current, they saw that the action potential, the end plate potential which is generated, it dies out slowly. So they introduced the electrodes over the certain distances, and they saw that the strength of the end plate potential is decreasing, and that is why end plate potential is a graded potential. It dies out eventually. So here you can see that on the muscle fiber, the inward current is generated, and this inward current travels through and then it leaks through the membrane. Okay, so this is leakage. Okay, this is leaking through the membrane. So this is creating small electrical circuits. These are the electrical circuits. And how the electric what is the direction of the flow of current? That is inward. Okay, let me zoom this picture a little bit. Okay, so this is how the current is flowing inward, and it travels through over certain distance, and then it leaks through and it creates a electrical circuit. So this is how it is, traveling over the distances. So the current is inward. So with the distance, the with as the distance increases, the current flow also decreases, because of the, because the current is leaking here. That is why the current eventually dies over the long distances, because it is not an action potential which has a continuous conduction. It has decremental conduction. So end plate potential shows decremental conduction. This also proves that it is an graded potential. It is a graded potential. Okay, so here, here is the schematic to show how the end plate potential is measured. So they have introduced micro-electrode to measure voltage and to measure ion current. So they are showing the this VM. This is measuring the membrane potential. So once the motor axon is stimulated here, okay, at this point, the membrane potential increases. Okay, so membrane potential is increasing, means the charge, whatever charge separation was there, it is decreased. So this is showing the depolarizing end plate potential. Polarized, something is polarized means it has two poles, two ends. So a membrane is polarized means it is going to have two poles. So one is positive end of the membrane and other end is negative, and that is why we are calling this membrane as polarized. So depolarization, D means something is being reduced. So depolarization means the whatever polarization is here is it is decreasing, means the charge separation is decreasing. So how decrease in charge separation means, the compared to this, the inner side is getting positive and outer side is getting negative. So let's say that the membrane potential here was minus 90 millivolt, and depolarization means decrease in this charge separation will mean that the negative value is decreasing here, means it is getting to, let's say, minus 50 millivolt. So this is how the end plate potential is the end plate potential that is created here. It is the depolarizing end plate potential. Okay, so this is how it is showing that it is depolarizing. Well, here the potential is positive. So like from my, let's say, ion flux was enough to create a positive potential, membrane potential. So here it is plus 10 millivolt. Okay, so and the IM, this is measuring the current. So once this is measuring current in nanoampere. So you can see it is showing 10 nanoampere. So this is showing that the whatever current was generated, that was inward. Okay, so this is showing the inward current generation. So with this, I end the lecture. In next lecture, we will learn about how the, okay, so, well, so this was a long discussion about neuromuscular junctions. In this lecture, I have already covered the structural parts, how a neuromuscular junction looks like and how end plate potential is created. In next lecture, we will learn about how the end plate potential, which is generated through a single acetylcholine receptor looks like. So in next lecture we will learn about the change in membrane potential that is seen through the single receptor, okay, single acetylcholine receptor. And then there will come next part, the third part of this lecture. In that lecture we will learn about the molecular properties of acetylcholine receptors, and then we will be done with the neuromuscular junctions. So this was a long discussion, and that is why I have divided it into the three parts. Okay, so we'll meet again in next lecture. Till then, see you. Bye bye.